Hello. So, I just thought I would give you a little introduction to what we're talking about in Chapter 8. Sometimes the overall concepts are just a little hard to wrap your head around. So, I'm hoping that this just makes Chapter 8 a little bit easier for you. If you're already comfortable with genetics, you can skip this introduction and just go right into the Chapter 8 Part 1 video. Okay, so in this video, we're going to talk about why DNA is important. We're going to talk about the leading strand and the lagging strand in DNA replication and semiconservative replication. So those are the big concepts for this little video. So DNA is the genetic material of an organism. It contains all of the genes. So if you have a gene for blue eyes or for brown eyes, for curly hair or for straight hair, if you have genes to make freckles or not make freckles, if you have genes to digest different sugars, that's all of your genes. So that is on your DNA, on your genetic material. Everything you can do and everything you're going to look like. That is true if you are a human being. That is true if you are a plant. That is true if you are an algae or a bacteria or a mushroom. Your DNA holds all of your genes, which is all of the instructions for what you can do and what that organism will look like. DNA is found in the nucleus of eukaryotic cells and in the cytoplasm of prokaryotic cells. All cells have DNA, and so for a cell to divide, whether it is going through mitosis or meiosis or budding or binary fission, anytime a cell divides to make a new cell, it has to copy its DNA so that a copy of the DNA is in a new cell. So when the cell divides, it's going to copy its DNA and make two identical copies of DNA. If everything goes right, you'll have two identical copies of DNA in both cells. One thing that's important is to notice that the two strands of DNA face different directions. So if you were looking at this picture of the students on the right side, they're facing different directions. So if they let go of their hands and start walking, they're both going to walk in the same, like they're both going to use their legs, they're both going to be doing the same things, but the two rows are going to walk in different directions. So even though both lines are made of the same kinds of students, they both are able to walk, they walk in opposite directions just because of the way they're facing. In DNA, you can notice this strand has the oxygens pointing up. So this strand is going 5' to 3'. So if you number the carbons, you start with 1, 2, 3, 4, 5. The little illustration shows you the carbon numbers. The 5' carbons on top and the 3' carbons on the bottom. So this is going 5' to 3'. On this side, the oxygens are facing down. These O's, those are oxygen, they're facing down. And it's going 3' to 5'. So these two strands are facing opposite directions. So when the DNA separates into two strands to start copying themselves, these two strands are going to have to move in opposite directions to copy themselves. So because they're moving in opposite directions, we're going to see very different things happening. So here, as the strands are opening, they're being separated by one enzyme. You have a primer that's going to come in and start it, and then DNA polymerase starts copying DNA. Here on the leading strand, it can just keep following helicase, and it's going to go across and make one long, continuous piece of DNA. That's the leading strand. The opposite strand is the lagging strand. Things work a little bit differently on the lagging strand. So as DNA starts to get separated, DNA can't copy itself in this direction. It has to go backwards. So you're going to have a backwards piece made, and then it gets to the end. So it has to come back and make another piece of DNA. Then come back and make another piece of DNA. This is the lagging strand. You end up with a bunch of little pieces of DNA. Those little pieces are called Okazaki fragments. Those little fragments then have to be sealed together to make one long strand of DNA. So in the end, you have two finished strands of DNA, a leading strand and a lagging strand, that have both been made using the same enzymes, but because they move in opposite directions, they're being made very differently. Another thing I want to mention on here is this is semi-conservative replication. So at the end, these two original pieces of DNA, you're going to have one strand going to each cell. So an old strand going to this cell and an old strand going to this cell. And you have a newly made piece of DNA here and a newly made piece of DNA here. So that's semi-conservative replication. You don't have one DNA getting two black strands and one DNA getting two purple strands. They're both getting an old black strand and a new purple strand. That's semi-conservative replication. So just to make sure you understand what we talked about, make sure you understand what DNA is, how are the two strands of DNA different, and what is semi-conservative replication. So those three questions you should be able to understand before you start on the Chapter 8, Part 1. Okay, see you in a little bit. Bye.